So lots of topics to cover today. So we're going to recap a little bit what we talked about in terms of DTI. We're going to talk about tract-based spatial statistics, get into fiber tracking, crossing fibers, and then really talk about sort of how sort of the modern, what the modern sort of protocols look like, and then talk a little bit about probabilistic tractography, and then we'll end with some of sort of the basic processing that you need to do for DTI. Okay. So as we talked about last time, at least for the very standard DTI protocols, what we're estimating is we're trying to estimate the shapes of these diffusion tensors. Okay, and especially in white matter, they're very anisotropic. They have a sort of a principal direction that's very clear that follows the direction of motion of diffusion. And then in areas sort of gray matter and CSF, you have much higher diffusivity and much more isotropic diffusion. And so there, the diffusion tensors are represented by spheres. All right. And so the two main maps that you'll typically see are there's always sort of going to be a fractional anisotropy map, which tells you how anisotropic the diffusion is, and there's going to be a mean diffusivity map, which tells you what's the overall diffusion. Okay, and as we noted last time, the diffusivity is highest in the cerebrospinal fluid. Okay. This is just another view showing very anisotropic diffusion in white matter. Fairly isotropic diffusion in gray matter, but much less diffusivity than there is in CSF, which is here. Okay, so these are fairly isotropic, and the size of the spheres is indicating that there's just higher diffusivity. So the size of the ellipsoids is typically used to represent how much diffusion there is, and then the shape tells you how isotropic or anisotropic diffusion is. So one thing that we're going to talk about is different ways that people have come up with over the years of how to display all this information. There's a lot of information to take into account. So one thing is that for every voxel, there will be a principal diffusion direction. So that's basically the first eigenvector. So that for and so for white matter, that actually has a lot of meaning because that's the direction of diffusion. For gray matter, not so much, and not so much for CSF either. Okay, so this really only has meaning in most cases for white matter. So this is one of the more common ways of displaying this. This is what FSL will do for you, and the end is you're going to end up with this colored map here. So we're going to just walk through what that color map means. Okay, so for every voxel, there's going to be like an X, Y, Z component of the first principal eigenvector. Okay, so it's going to just, on one map, it's going to take the X component, and this is where there's a lot of X component in the diffusion. There's a Y component, so this represents like diffusion going up and down, and there's a Z component. So you literally, for every voxel, you just take that vector and you plot its X, Y, and Z components in different maps. Okay. The next thing we want to do, though, is we want to say we only want to look at those voxels or really highlight those vectors where there's lots of anisotropy. Okay, we don't really want to have to look at those voxels that are isotropic anyways, because the direction doesn't really matter for those voxels. So then we're going to multiply it by the fractional anisotropy map. Okay, so that sort of downweights those voxels that are fairly isotropic. And then, so the next row is what we end up with. And then what we're going to color code each direction. Okay, so the red channel is going to represent sort of the left to right diffusion. The green channel is going to represent the A to I, and the blue channel represents the S to I, or the inferior superior. Okay, and then we just sort of merge all those into one color map. All right, and so that's what the color map looks like. And so then obviously where those colors sort of merge, like if you have some blue and some green, that means that there's going to be something going in the XY direction, right, as opposed with pure X and pure Y. So this is an example of that color map, and so you can sort of see here, where it's clearly directions here. This is very green, okay. Left to right is very red here, but in between you sort of see a mixing of colors where the voxels aren't purely X or Y, Okay, so this is sort of what's called a color-coded orientation map. Right? So I mean, they're very pretty pictures at the very least. So you should definitely put that in your publication or in your thesis, right? And then in terms of applications, it's also a very quick way of seeing what's different. So for example, here is a picture from a healthy volunteer on the left and a patient with dysplasia on the right. And clearly, they're basically, this thing that's in the patient appears to be missing. That's in the healthy subject, that sort of anterior posterior fiber appears to be missing in the patient. Okay. So it's just a very quick way of seeing what's normal or what's abnormal. All right, any questions on that? Okay. So the next topic we're going to cover is tract-based spatial statistics, which is developed by the folks at FSL. And the idea is, if you want to do statistics on the diffusion, how would you go about doing it? For example, if you want to see what areas of the brain might have different fractional anisotropy, how would you compare a patient population versus healthy controls? Okay. One of the challenges is that the fibers don't all run aren't perfectly the same. If you just did the anatomical and did the anatomical registration, there's no guarantee that all those fibers are going to line between your subjects, right? So then the problem is then if you're looking at the FA in a voxel, sometimes you'll be hitting the fiber, but in another subject you're hitting gray matter. And so you're going to be really confused by what's going on. Okay, so the way they do it is that they sort of deform everyone anatomically. Okay, then they take the FA maps and they sort of overlay them on top of each other and create a mean FA map. Okay, and then they use that to form what's called a skeleton. So this is sort of like averaging all the subjects and then finding out, where on average is the fractional anisotropy greatest. Okay, and then they can threshold that to sort of form a basic skeleton of saying, well, we think that, if we're going to do comparisons, we're going to do it along this skeleton, okay? And this skeleton forms sort of the reference point. And so this is a picture of that skeleton where you're seeing in blue here the skeleton overlaid on the mean fractional anisotropy. And you can sort of see that skeleton, it sort of falls right in the sort of the middle of the, sort of the ridges, the peaks of that fractional anisotropy. Okay, so it's trying to sort of say, on average, where do we expect to see, where do we want to measure the fractional anisotropy to compare between subjects? And then what you do for each subject is you take that skeleton and then you look in each subject's FA map and find the sort of the closest peak. Okay, and so you're sort of, even though the skeleton might be here, maybe this subject's FA map is, strongest over here. So you're sort of grabbing the value that's closest, that's in the mean, sort of the peak of this FA map, and bringing it to here. Okay, and it's sort of a way of sort of getting all these different FA maps from different subjects aligned to one common reference frame. Okay, so then for each voxel of that skeleton, you're going to have different FA values from all the different subjects. Okay, and so then you can start asking for that value in the skeleton, for that position in the skeleton, is there, for example, a difference in FA between, say, normals, patients and controls? Okay, so it's not necessarily the only way of doing it, but it is the way that FSL has chosen to do it. All right. So then what you do is essentially you have all the values. So every subject now has their values sort of morphed onto this skeleton. And so now you can compare the values on that skeleton. Okay, and so for example, for every voxel, you can have a general linear model and have all those FA values between controls and patients, and then do t-tests or whatever you want to do. Right? So that's the basic idea. So this is example of TBSS results looking at ALS patients, and the red is showing where the FA correlates negatively with ALS progression rates, and so that's would be done with GLM, where they've projected the FA values onto the progression rate. And then the blue is showing where FA is significantly lower in ALS than in controls, which is basically almost everywhere. Okay, so showing that in ALS patients, there's much less fractional anisotropy. Okay, so does anyone know why the ALS patients would have less FA?
Do they have less myelination?
Right, they have less myelination, right? Okay. Okay, so that was a fast zoom through TBSS, so we're moving right along. So any questions? Okay, so now we're going to talk about diffusion tractography, which is sort of more of the, visually, one of the more stunning parts, of DTI. And I think this is a really nice map down here on the bottom, which is the tractography maps from the Human Connectome project, showing what you can do with 8.4 minutes, 11.5 minutes, or 25 minutes of data. And I think it's still remarkable that you can put a human being in a scanner for 8 minutes and come up with a map like this. It's pretty remarkable. Okay? And there's really no other option, right? I mean, there's no other way to do this, right? I mean, you could inject tracers into people, but you'd have to do it for every fiber. You could wait till they die and cut them up, but it's still really hard to do. So this is, and it's an in vivo method that works really well. Okay. So the basic idea behind fiber tracking is fairly simple. Is essentially you just follow the direction of the arrows. Okay. So for every voxel, you have a principal direction, and you just sort of connect the dots. Okay. There's obviously different ways of doing that. There's ways of, how do you sort of, do you just do nearest neighbor? How do you interpolate between voxels? We're not really going to go into those details today. I mean, there's just, that's more of a technical detail, but the idea is that it depends on having a principal fiber direction in each voxel that you can trust. We'll talk, so a lot of this lecture is going to be talking about when that assumption fails. Okay, what do you do? So typically, now if you just do something like that, then you end up with really weird tractography. Okay, if you just follow those things, and so typically these algorithms will put limits on what's good and what's bad. So in this case, they put a FA threshold. So a voxel would have to have an FA of at least 0.2 to be considered as part of this tracking, which makes sense because if you don't have any fractional anisotropy, it really doesn't you can't really trust that direction. Okay. The other thing, now they've added this threshold, and they've also added a minimum radius of curvature, meaning that you can't have paths that go like that. Okay, so it's just trying to get rid of really things that we don't really think happen, but they may happen in some areas of the brain. But in general, these algorithms will put different criteria on it. Okay. And if you do things like that, then you can end up with really nice tracts here, showing sort of using DTI to come up with 9 sort of major fiber tract families. Okay. Here's some really nice tractography showing how the fibers develop with development, okay, from 2 weeks to 1 year to adult. So now we get into some of the downsides with tractography, and this is one of the reasons that has led to sort of the more modern imaging protocols, is the issue of crossing fibers. Okay, so in the brain there's a lot of crossing fibers, and so this is something that people have been trying to work on for quite a long time. So what's the problem with crossing fibers? Well, so if you just have, here we're showing there's just a very one fiber orientation, and so for that one, if you do DTI, then that's fine. You end up with a diffusion ellipsoid which points in the same direction as the fiber. Great. If you have all the fibers aligned this way, then DTI works really well too. Okay. But let's say here where we have fibers running this way and this way, right? So now the ellipsoid is going to have sort of 2 equal principal directions, right? So it's really not going to give you much information about which way the fibers went. I mean, I could have had fibers up and down or fibers at any orientation, and it would still give me the same information in terms of diffusion ellipsoid. Okay? So that's something, if you're trying to track something and you come to one of these voxels that has crossing fibers, you don't actually know which way to go now. Okay? So that's been one of the things that people have been working on over the years. And another thing you'll notice that it's also something that you have to consider when you're looking at your fractional anisotropy maps. So even if you're not doing tractography and you're just looking at FA maps, you need to be careful about how you interpret differences in FA, because notice here, these places where the FA is low, or I guess here, these places. Places where FA is low, it's not necessarily that the FA is low, it's just you may have crossing fibers, right? So for some reason, let's say there was more crossing fibers in one population than another, that would be something you would need to consider. Okay, and so that's where DTI sort of lets you down, because it doesn't, if I only have, say, a B equals 0 image and then 6 directions, all I can estimate is this one ellipsoid. Okay, and so I'm lacking the information I need to differentiate these cases, which actually could have an impact on how I interpret my data. So this is just another model showing, in certain areas here, the DTI does really well. So here, the first eigenvalue is much greater than the other two eigenvalues. So there's a clear direction of diffusion. Okay, so you can feel fairly confident of interpreting what's happening there. But if you look here, there's a spherical tensor here with all the eigenvalues the same. Okay, so what do you guys think is happening there? Like, what could give me a spherical tensor at that point? It looks like there's a lot of white matter there, right? And how many fibers do you think are crossing there? A lot, at least 3, right? So one this way, and one this way. So there's 3 principal axes, and they're all the same. So, if you see a spherical ellipsoid, a spherical tensor in an area where you think there's got to be fibers there, then there's at least 3 fibers there. Okay. What about here? This oblate tensor, where I have lambda, the first 2 eigenvalues are about the same, and they're much greater than the third eigenvalues. How many crossing fibers do I have there? 2, or at least 2. There could be more than 2, but they're all in the same plane, right? There's definitely not one in this other plane. Okay, so it's at least 2. There could be more than 2, but they're all crossing in the same plane, as opposed to all 3 planes. Okay, so as you're looking at these FA maps, realize that the FA maps in certain areas are telling you about the true anisotropy, but in other areas, if it's purely based on DTI, then it's not really telling you the whole picture. Okay. This is just another picture, just trying to emphasize that if you, for example, you see an oblate tensor, that you can't really interpret it as just one direction of diffusion. Okay. So when it's pancake shaped, that's typically called an oblate tensor. Okay. And so just to remind you, it means that there, the principal direction is not necessarily the direction of the fibers, and FA changes are difficult to interpret. All right. And this is a big problem. I mean, it happens a lot. So this is just a plot showing the regions where, so red are those areas where, here, where there's just one fiber direction. Green is those areas with 2, and blue is where there's 3 fiber directions. Okay, so a lot of the brain has a lot of crossing fibers. Okay, and so this is just showing sort of the volume fraction where there's more than one fiber, and as you can see, it is, in some cases, it's like 60 or 70% of the volume fraction. Okay, so we talked a little bit about how crossing fibers can affect your FA maps, right? Obviously, they can also affect your tractography, right? Because if I have, let's say I actually have 2 crossing fibers like this, right? But if I don't know that, I might interpret the fibers going like that. Okay, so how do I distinguish between those two? Okay. Today we're going to mostly focus on what FSL does, and mostly in the sense that that at least gives you a background as to what the problem is. Just know that diffusion is a very controversial and active field. Everyone has their own solution, and so you will have to just decide what you feel works best for you. But FSL is probably the best starting point, at least for an open source solution, and then you can get more sophisticated from there. Okay, I think Alan will probably talk about some more sophisticated solutions or point to them on Monday. But this is just an example showing that in this case, this fiber went this way where there really shouldn't be that connection. and it also missed, it really should have gone this way because there should be a connection that way based on what we know about anatomy. Similarly, you know, the connections that went this way should have gone this way, okay? So where the fiber tracking, just because there was some confusion about where it should go, it's like you're at an intersection and you're not sure which way to go, and you just go the wrong way. Okay, so there's only maybe one question today, so let me see if we may not do Canvas because I didn't remember to set it up, but if I do. So go ahead and think about it, and maybe we just do this old-fashioned way. Yeah, let's just do this the old-fashioned way because otherwise I've got to do my duo push and all that. Okay, so just if you guys can take a look at this. So basically what we have shown on the right-hand side are 5 different pictures of fiber orientations. Okay, and then on the right-hand side we have 5 different sort of particle distributions. Imagine particles diffusing in that structure. And the idea is to sort of think about which of those particle distributions line up with which fiber configuration. Okay? So just take a minute or so to think about that, and then we'll just go through this together. Okay, so let's take a look at A. Which distribution do people think goes with A? 5, yeah, okay. So 5 is sort of got the strongest principal direction, and that's similar to what we've been seeing before. Okay. What about B? Right. It could be 1 or 3, but 1 is pretty good. Okay.
1 and 3 or 1 and 4?
Well, 1 and 4, right? They're basically almost look the same. Yeah.
Is there any meaning in the fact that they're like shifted?
One is called fanning fibers, and the other is bending fibers. But you sort of see they're going to give you the same problem, right? You got 2 sort of directions emerging like this, or you've got 2 directions this way. So in that case, it's a little hard to tell because over that voxel, this versus this is basically, if I have, this is like bending fibers, right? And this is that fanning fibers, and this is bending fibers, right? But you can sort of see over the course of a voxel, I could just take, if I split that in half, I could take this bend and put it up there, and it'd be a fanning. So it's going to be very similar results. Okay, and there's a lot of that stuff happening in the brain. Okay, how about D? Which would D go with? 2, right? Because those voxels are sort of crossing like that, right? Or those fiber bundles. And then by process of elimination, E must go with 3, right? Okay, and they're crossing perpendicularly. Okay? Question?
Are any of these kissing fibers?
Kissing fibers. No, kissing fiber would be like this. Well, the bending is close, but not quite. So kissing fibers would be like that. That's the kissing fiber. Okay. Good question. What do you got? What do you think?
Does it look like a straight line?
All in the same voxel?
Yeah, let's say they're all in the same voxel. Let's say this is all in the same voxel. What do we think that the distribution would look like?
Kind of dispersed? Or?
Dispersed. But let's make it like this. What would that look like?
Do they cancel?
Can you distinguish that from this? Not really, right? Because the MRI doesn't know what, I mean, you could probably, with some more sophisticated things, but at first order with a DTI fit, it wouldn't really distinguish that. Because assuming that the diffusion was equal in both of those, right, then the stuff that sort of goes like this will somewhat mimic the stuff that just could have gone across. Okay? So to first order, yeah, kissing fibers would appear as crossing fibers, which is a big problem, right? Because you're tracking them. You're supposed to track them this way, but you're tracking them this way. Okay? Any other questions? Okay. So the solution for that is what's called high angular resolution diffusion imaging, and this was actually part of the very early work on this was done by Larry Frank here at UCSD, and so this goes by the name of A.R.D. Okay, and typically what it means is we're just going to do a lot more directions. Okay, instead of doing just 6 directions, we're going to do lots of directions over the sphere. So we're really going to characterize what that diffusion looks like over a very high angular resolution. And so typically we'll go to 60 directions or 100 directions. Okay. And then the idea is that what the field has decided, or at least what FSL has decided, is to say that, well, to really do that, if you characterize that, there is a way to go back and fully characterize the diffusion, but it just takes a lot of measurements to do. And so what the field has come up with is more of what's called a model-based approach, where let's assume there's 1 fiber or 2 fibers or 3 fibers. What would the diffusion signal look like? And let's just see what's the best fit for every voxel. Okay? So here's the basic idea. Let's say I have 2 crossing fibers. Okay, so that's 2 fibers that are crossing. I say, well, the diffusion signal from a fiber going this way, the diffusion signal looks like this. So that's the diffusion signal. And let's think about that a little bit. Why does the diffusion signal look like that? It means if the diffusion is along this direction, if I put my gradient along that direction and measure, I get a very small signal, right? Because there's a lot of diffusion in that direction, so the signal is going to decay away very quickly. If I measure the diffusion in another direction, that way I get a big signal because there's not much diffusion in that direction, so the signal doesn't decay away. And then for any other direction, I get sort of a fall off. This is actually, you're going to find what's called a cosine squared theta fall off. Okay, so the idea is if anytime I have, let's say this is my fiber here, right? And I measure along this direction, I get very little signal because lots of diffusion decay. If I measure in this direction, I get a very big signal, right? And then anywhere in here, I get sort of a fall off of the signal. Okay, so a single fiber or fibers going all in the same direction have this characteristic peanut shape. Okay, we actually call them peanuts. Okay, and so the idea is if I have 2 fibers, then I just add those 2 peanuts together, and this is what I expect to see. Okay, and that's all it is. That's my model. If it's saying if there were 2 fibers, then that would be the diffusion signal I expect to see. Then the trick now is for every voxel, you have to figure out how many fibers there are, and then what's the angle. Okay, so you might say there's 2 fibers, and I think the angle is this, or there might be 3 fibers, I think the angle is that. So it's this big fitting thing where you're looking for what's the best fit to your data. And so that's basically what FSL does. So if you use what's called bedpost X, and I have no idea what that stands for, they have the weirdest acronyms, and this is one of them. Their model is this, that every voxel has an isotropic part, which is a sphere, and then it's got at least one, or it can have 1, 2, or 3 sticks in it. Okay? And so this is called the ball and sticks model. Mathematically, it's represented this way, where you have F is the volume fraction for the anisotropic part. Okay, so if there's just, we're saying some of the signal comes from that anisotropic part. The remaining part, which is 1 minus F, comes from the spherical part. So that's 1 ball, 1 stick. Okay? And then you can expand that into, now we're going to ignore the ball part for now. We're just going to talk about the stick part for now. So you can expand that and say, well, what if there's, I'm not sure of how many sticks there are. Maybe there's 1 stick, maybe there's 2 sticks, maybe there's 3 sticks. So for every box, I'm going to open up the possibility that I need to figure out which is the best fit for the data. And the idea is if the measured data looks like this, I'm going to say it's 1 stick. If it looks like that, it's 2 sticks. And if it looks like that, it's 3 sticks. Okay? And then And then each stick can have its own volume fraction. And then I just sum them all up, and I'm left with, this is the anisotropic part, and this would be the isotropic part. So the full model is there's a ball and 1, 2, or 3 sticks. Okay, and each with a volume fraction associated with it. This is just showing you a little bit of how things look. So for example, BD stands for the B value times the diffusion coefficient. So this, for example, the red here is for a fiber going this way, and the green here is for a fiber going that way. Okay, so B times D is, in this case, it's 1.5. Then those look pretty much like peanuts, and they look like pretty fat peanuts. Okay, and then when I sum them together, you can sort of see that I end up with something that's almost like a square. And it's going to be a little hard to figure out the diffusion direction from that, right? Because if there's any noise here, right, it's going to be sort of hard to really tell what that shape is and figure out which direction things are. In this case, I've made B times E equal 4.5. So in this case, I've increased the B value. And now you can sort of see those peanuts that got a lot squished down. So now when I sum them up, it's really easy to see the directionality, right? So even if I add noise to this, right, I'm still have a good chance of detecting the directions, right? So this is one of the reasons why you sort of see large B values and the field incorporating more high B values, and we'll talk about that in later parts of the lecture as well. All right. Okay, so that's the basic idea, and so then it's fairly simple. So if I have 2 sticks, then the measured signal is in blue. DTI would predict a circle, which is clearly wrong. The ball and sticks fits the data much better. Okay, if I have 3 dimensions and I just sort of look at it from the side, then do a cut through, then DTI once again is going to try to fit it with a circle, which is saying it's isotropic, and the ball and sticks does a better job of saying, no, actually you have 3 fibers there. Okay, so that's the idea behind what FSL is trying to do. Question?
I'm missing the intuition for why DTI can't do it.
Oh, because DTI can only fit things that are like ellipses.
Okay.
Yeah.
Oh, okay.
Yeah, it's just the model is just, it's got to be an ellipse. Yeah. And a circle is just sort of the best ellipse it can fit to that data. Yes?
What is the resolution of a stick biologically?
There's no, the stick just means it could be many fibers, but they're all pointing in the direction. Yeah, because whether you have 1 stick or 2 sticks or 3 sticks, as long as they're all pointing in the same direction, the diffusion signal has the same properties. Because they would all, it's the same shape, and you just sum them up, and it's still the same shape. Yeah. Okay, that's a good question. Okay. So then the problem is, and the hard part, which is what the FSL guys have solved, is the model selection. Is there 1 fiber, 2 fibers, or 3 fibers? And so there's a lot of math that goes behind deciding that. And they call it automatic relevance determination. Okay. So then at the end of the day, they basically sort of can you run in FSL, it's called bedpost X. And one thing that they'll do is they'll typically use a threshold about, let's say, 5% to exclude any sticks that have a very tiny volume fraction, right? So if the model says, well, 5% of this voxel might have a stick going this direction, in general, the feeling is, well, that's probably just noise, and so we're going to discount that. And so this is only sort of showing that in a lot of voxels in white matter, there's evidence for at least 1 stick. And then there is some evidence for 2 sticks, quite a lot, and that matches what we saw before in what voxels had at least 2 fibers. But then as you can sort of see, there's relatively few voxels that have enough evidence to support that there's 3 sticks. Okay? And this is just showing you sort of the display here where you can sort of see that for every voxel, it's either showing you 1 stick, 2 sticks, or 3 sticks. Okay, so here there's 3 sticks here, 2, 6 here, and in a lot of voxels, there's just going to be 1. And notice, because it also has an isotropic part, it's able to fairly handle well even those voxels that have a lot of CSF in it, right? Because it uses that isotropic part to sort of take out the CSF part, right? So it's okay, I can model all this with something that's isotropic so that it can still figure out what is not isotropic. Okay. So this is just to drive home the message about the difference between DTI and ball and sticks. So with DTI, you are limited to just one primary direction per voxel. With ball and sticks, you can have typically up to 3 sticks. Okay. The next thing we're going to talk about is multi-shell diffusion acquisition. So if you're familiar with either the HCP protocol or the lifespan HCP protocol or the ABCD protocol, you notice that they don't just use one B value. Okay, they typically use 2 or 3 B values. And so we want to talk a little bit about why we do that. And we already sort of saw that before, which is basically what we're showing you here is the patterns if you have 1 fiber, 2 fibers, or 3 fibers. And then the color scheme shows increasing B values. Okay, and similar to the slide we showed before, as I increase the B value for these crossing fibers, you sort of see that the shapes, the patterns are much better defined as I go to higher B values. Okay. And the basic idea is the higher the B value I make, the faster things that are in the diffusion direction fall off, right? So there's just much more distinction between things that are, so if this is the fiber and the B value is not very strong, then the signal this way versus the signal this way, there may not be too much difference, right? Because there's only a little bit of decay here as opposed to not much decay here. The bigger B value I make, this signal falls, I basically kill this signal. And so there's a very big distinction between this signal and this signal. And so your angular sort of resolution, your effective resolution becomes much higher with B value. Okay? But why wouldn't we just crank up the B value to like 1,000,000? Yeah, then you have nothing to measure, right? Then you're just measuring noise. Like if I crank up the B value to like, 20,000 or 30,000, I would actually have just sticks, right? The theoretical signal would be sticks, but the actual signal would just be noise because I have nothing left to measure. Okay, so that's shown here where as I increase the B value, you sort of see it's rapidly approaching noise. Okay, so then there's a question of, well, what's the right sampling of B values? How many small B values should you use? How many B values should you use at each angle? So anyways, these considerations have led, so this is A.R.D, so this is high angular resolution that lets you sort of, at least take a first stab at looking at these crossing fibers. And what the field has sort of moved to is what's called multi-shell diffusion, where we actually do several shells, and each shell is sort of sampled. Okay. But there's a lot of work and sort of simulations that go behind sort of determining that. And so this is some work from the Human Connectome project where they first looked at, like these are all, looks like 2 shells, these are all 3 shells, these are all 1 shell. And they're looking at things like, so this is the sensitivity to fibers, okay? And so they looked at sensitivity to 2-way crossings and 3-way crossings. Ultimately, they decided to look at sort of constrain the search down to here. And then they found that if they used B of 1,000, 2,000, and 3,000 with equal number of shells, equal number of directions per shell, they got their best performance for both 2 fiber and 3 fiber crossings. And so basically, this was the sort of the simulation or the calculation they did to come up with their protocol. And this was really the first sort of widespread multi-shell protocol that was used. But one of the problems was it took forever. It's like 53-minute long just to measure diffusion. Okay, so it includes 6 runs, each representing a gradient table, and then in addition, each was required in either the left-right or the right-to-left phase encoding polarity, which we'll talk about later when we talk about distortion correction. All right, so very great data set, but and acquired on over 1,000 people, but really hard to scale up to other populations. Okay, but if you're looking probably for the best diffusion data set that there is, this is still probably it, because it's just there's probably never going to be another study that spends an hour on so many subjects. Okay. So the next step down is lifespan ACP, where it's now cut down to 21 minutes, and they're going to do 2 shells, one at B1500 and one at 3000, with 92 to 93 directions per shell, plus 6 b equals 0 images per run. Okay, so that's a little better, and they're basically here. This is from, I forgot to add the reference, but they're comparing sensitivity between. This is the old protocol, so this is the old one, and this is the new one for the development, the lifespan project. And you can sort of see for most of the metrics, it matches pretty well. The only place where it really is not as good is for three-way crossings. The old protocol is still better than the new protocol, but they made that trade-off because for younger populations and older populations, they really felt like they had to cut down the scan time. Okay, so that difference in sensitivity is largely sort of attributed to the almost factor of 2, more than factor of 2 reduction in scan time. Okay. And then the other major protocol that's out there now is the ABCD, which has now gone down to just 1 diffusion scan. And since it's meant for kids, it's even shorter, so just 7.5 minutes with 96 directions plus some calibration scans. So roughly 9 minutes total when you're done. And it uses 3 shells. And so this is sort of showing you a plot of there's stuff at B of 1,000, 2,000, and 3,000, and also some B equals 0 images. And this sort of lists how many are done at each direction. There's also done some at 500. Okay. So as you can sort of see, they made a different choice in terms of how to sort of sample the shells, and for different reasons. But the trade-off is also because they had to scan in kids, they're having to acquire much less data. Okay. But the thing that is consistent or common between all these different protocols is you see they all typically max out a B of 3,000, okay, and they're all doing at least 2 shells, okay. And so the idea is the field has decided that B of 3,000 is a pretty good sort of maximum operating point where you're not losing all your signal, you're still getting enough information that it makes sense to acquire it, okay. But it still helps to acquire data at lower B values to get back some SNR where you don't need to have that, a high B value sensitivity. And then finally, and so bedpost can do multi-shell, and one other thing it does now is it takes into account the fact that diffusion depends on B value, which we haven't really talked about before. Okay, so previously all we've talked about is diffusion is proportional to like e to the minus b times the diffusion factor. Okay, so we've said that there's just this exponential decay of the signal with diffusion as a function of b value. Okay. In practice, and so if I took the log of that, I would get a straight line. Okay. In practice, and this is very important for the brain, what you find a deviation from a straight line. So actually, the curve actually goes away. It doesn't decay as fast as you think it should. Okay. And what's happening is, typically in every voxel, you're going to have a mix of different populations of either different tissues, or some water is in the cells, some water is outside of the cells. They all experience different microstructure in the brain. Okay. Some of those particles are going to be very fast diffusing, right? And if that's if that's most of your volume, then you're going to have a very straight. Initially, you're going to fall off very quickly with the B value. Okay. But then once that signal is gone, then you have these more slower diffusing voxels. Especially, for example, let's say something's trapped in a cell and is slowly diffusing, then now you're going to start to see those signals more. Okay. So in every voxel, you actually have a distribution of the rate of diffusivity. Right. It's not just that every voxel there's just one diffusion coefficient. In any given voxel, there's probably a big range of diffusion coefficients. Okay. So the next level of modeling is taking that into account. And so that's one way of doing that is something called diffusion kurtosis imaging, which some of you may have heard of, which is basically trying to, the data actually looks like this, DTI and most models will fit just a straight line, and diffusion kurtosis imaging is trying to model the fact that the diffusion doesn't happen as quickly as you think it should. Okay? So for example, if it was just pure free diffusion, the particle distribution would look like a Gaussian. In practice, what we see is things don't fall off that quickly. Particles are sort of trapped, and so the diffusion isn't as fast as we think it should be. And so just to give you a sense of what that means is that this is sort of what for different b values, what the signal looks like, the MR signal looks like as I'm increasing the b value, and sort of showing that, what we've seen before, that sort of that peanut showing up. The problem is because they're modeling a distribution of diffusion values, it turns out that even though this is a single fiber, if I do, so this is, I think, let's see, 1, 2, 3, and 4 fiber fits. The data is actually fairly well fit by 3 fibers, possibly even better fit by 4 fibers. Okay, so the idea is if you have multiple diffusion compartments, that can appear to be multiple fibers when actually there aren't multiple fibers. Okay, if I just had like 1 fiber, but I had some of the stuff was trapped and some of the water wasn't trapped, then it could be modeled as multiple fibers, and I would make the wrong conclusion about what's going on. Okay, so in general, if you don't model this distribution, you're going to overestimate how many fibers you have. Okay, so that's what bedpost tries to do. So this is shown here where, let's see, what is this for a second, and blue third fibers. Okay, so this is the mono-exponential. So this is, how many fibers would have, how many voxels have 2 and 3 fibers in it. Okay, so that's if you didn't take into account the fact that there's different diffusion compartments. If they do what's called this gamma distribution and sort of say, well, let's go ahead and take into account the fact that there's going to be different diffusion properties, then you can sort of see the number of voxels sort of that have, so the mean volume fraction of voxels that have 3 sticks is much less. Okay, meaning that if you just model it this way, you're going to overestimate how many sticks you have. Okay, so that's just another place where, as the field advances, we're sort of getting better and better at trying to estimate, what's actually going on in terms of diffusion. Okay. Any questions? Okay. Okay. So then once we have those multiple sticks, then we can apply tractography, and now we can sort of better try to resolve crossing fibers and. But there is still the question about the fact that our measurements aren't perfect. Okay, so let's say we just came up with a tractography from a single data set. If we just took that same subject and repeated the experiment many, many times, each time we do it, we're going to end up with slightly different fibers, right? In fact, if you just overlaid all the different fibers on the times you did that, you'd end up with sort of only evidence for 1 or 2 tracks, not all these tracks. Okay. So in FSL, the prob tracks X implements what's called probabilistic tractography to try to take this into account. Basically saying that in every voxel, there's some uncertainty as to which way the voxel is going to go. So basically, instead of just coming up with one path that might go like this, it comes up at every voxel, it just sort of says, well, the data suggests I could go anywhere, any of these, within some angle of paths. And so it just randomly picks one of those. Okay? And so it comes up with lots of different possible paths for you. Okay? And then it tries to estimate the different probabilities of the paths. All right? And so, for example, if I hit a voxel like this, so what does that mean? What kind of diffusion is happening in that voxel? Isotropic, right? It could go in any direction. So at that point, if I hit an isotropic voxel, it can pick any direction to go in. Okay? So it just basically uses the computational power that we have now just to run lots and lots of streamlines. Okay? And so then it can basically, and then the other thing it does is that if there are multiple fibers coming in one voxel, it basically picks the one, and as something's coming in a certain direction, it just picks the one that's most compatible with that direction. Okay, so there are some assumptions built into it, but in general, it's just trying to take into account, admit that there is uncertainty in the data. And so what it comes up with now is for every sort of tract and every position, it has a probability of whether there's a tract there or not. Okay, so instead of just saying here are the tracts, it's trying to say, well, this is how probable that tract is. Okay, and so what you can do with that now is, so you do all this tractography. So the next question is, what are you going to do with all that data, right? How do you actually do science with that data? So typically, one thing you might do is you now want to come up with connectivity matrices. Okay, and so what ProbTract will let you do is it'll allow you to compare, ROI to ROI connectivity, or voxel by ROI connectivity, or voxel by voxel connectivity. So it allows for all these different scales of connectivity to look at. Okay. And the way it defines connectivity is basically, let's say I want to look at the connectivity between A and B. Okay. Basically, it looks at how many streamlines come out of A, how many possible paths come out of A, and says how many of those paths went through B. So that's one way of defining connectivity. Okay. And that's using the probabilistic approach. So that's the probability of starting at A and ending up with B, and so that's what they're going to call connectivity. So the resulting connectivity, for example, between two target ROIs could just be, what's the probability of starting in any voxel here and ending up in a voxel here, or starting here and ending up there. Okay, and so you could come, if one of your hypotheses was, how does connectivity between region A and region B differ between populations, then you could use ProbTract X to sort of estimate that connectivity.
Look at that within subjects as well? Or
You mean with
task conditions?
Well, yes, you could. For example, I mean, it would have to be a task condition that was strong enough to change the connectivity of diffusion. So what you could do is, for example, measure people before and after. Like there was a very old study of looking at, juggling for 3 weeks, right? So you could look at the effect of an intervention that way. But within a certain scan, whether you would see differences in diffusion. There's debates about whether diffusion changes with function, and so there is an area called diffusion fMRI, which is arguing that you can use diffusion to detect actually very quickly changing changes in cell swelling due to function. But that's still very controversial whether you can do that. Okay. And I'm not sure, maybe someone in this room knows in terms of is there any, in terms of how quickly you can change diffusion, the structural diffusion connectivity. Does anyone know is there any convincing study that shows you can do it within a day or an hour or, okay. And we do know that, I mean, plasticity happens very quickly, so I just don't know what the sensitivity is, yeah. Okay. This is another study where they basically looked at, for each voxel in the thalamus, they basically looked at which voxel in the cortical area showed the greatest connection with that voxel. Okay, and so they were able to sort of then partition the thalamus into the different brain regions, and so saying, well, this area of the thalamus is most connected to this area in the brain, in the cortex, and so on and so forth. All right. Okay, so actually we're doing really well on time. So I think we'll probably be able to end on time. So the last part is actually just more sort of setting you up for, what will you do in practice. And so the FSL pipeline really has sort of two approaches. They all start off with the diffusion images, and then we'll spend some time talking about some of the pre-processing, top, up, and eddy correction. But then it sort of goes between, sort of there's two approaches. One is you can just do tensor fitting using DTI DTI fit is what they use. And then so you can do you know, fraction isotropy, mean diffusivity, and come up with all those nice maps. Okay. So why would you do this, given the fact that you know all the problems we've talked about with DTI? Like, why should you still do this? Time, right? This is really fast, right? The other approach can sometimes take hours and hours of processing. If you have a GPU, it's much faster, but you know, if you're trying to do it on your laptop, it might take a long time. So the upper path is still good to do because at least it allows you to see whether your data are reasonable, right? If you run it through this algorithm, which is very quick to run, and you end up with just noise, and you know, oh, something's wrong with my acquisition. Okay. So one thing is that I don't think we've really stressed in this course yet is all of you should always be looking at your data as soon as possible after you've gotten it off the scanner. And as soon as possible, if possible, like go, you know, once you're off the scanner, save your data somewhere, okay, make sure you have a backup, and then run it through your pre-processing as quickly as you can, especially if you're doing another scan the next day or something, because you never know. There could be something wrong with the scanner, there could be something that's going on with your protocol that you haven't identified. I mean, we've had people come to us like 2 years later and say, hey, I finally looked at my data and there's this artifact. And we're like, well, we really can't do anything right now. I mean, you know, if it's not too bad, you know, maybe we can help do some processing on it. But in general, you want to sort of take your data, and if you can go all the way through the analysis, then that's even better. Because sometimes you may not see something until it's far in the analysis, and then something looks just a little bit off. Okay, so for diffusion imaging, definitely run it through till you look at you can look at the FA maps, and if they don't look reasonable, then you need to sort of, you know, say, hey, something's wrong. Okay, and then if you have more time, then you can run the bedpost and the tractography and all the more sophisticated things. But you definitely want to do DTI fit, you know, sort of do your diffusion tensor imaging analysis very quickly, and then you'll be satisfied that hopefully at least the data is reasonable quality, and then you can do the other more sophisticated analysis later. Okay, so if you're just doing that very standard analysis, then the steps are top up, which we'll talk about, A.R.D, which we'll talk about, and then DTI fit is what actually does the imaging. And then, you know, if you want to then do the group analysis, then you would register it and, you know, do your tract-based statistics. But for per scan, you should at least be doing these three things. Okay, because if your data doesn't look good after those three steps, then there's something's wrong. Okay, so just sort of note on good experimental practice. Then if you want to go on, you can use bedpost, which sort of does the multiple shells, the multiple directions, the ball and stick model. So this will do this sort of the ball and stick model for you. Okay, and then the prob tracks will then do the probabilistic tractography. And then Alan will talk about other things you can do with the data as well. All right, so there's really sort of a first stage of analysis and then more complicated analysis, which you can take your time with. Okay, so we're going to spend the last 15 minutes talking about things that you generally need to correct with your data. And one of the things is, which we talked about before, is that anytime you put someone's head in the scanner, there's going to be significant field inhomogeneities, especially in the orbital frontal cortex where you have the skull-air interface. Okay? And what that means is that because of the way the imaging is done, and we talked a little about case space, and so you'll hear about things like AP versus PA, okay, and it's like, what the heck is that? So the idea is, in the Fourier domain, it depends on whether you go through case space like this way or this way. That's all it means, okay? So it's how we traverse Fourier space it actually affects the image acquisition, okay? So in this case, if we go through Fourier space one way, it sort of smushes the front of the head. If we go through the other way, it stretches the front of the head. But those are do the same thing. And the fact that it does it in opposite directions is actually something that people figured we could take advantage of. Okay. So that's one source. So one source of, and the major source of that is, you know, if this is your human head, a CT, the resulting field is very different in the orbital frontal cortex and also around your ear canal. Okay? And that leads to that distortion. This is particularly nice demonstration of how it really gets smushed in. So the amount, each voxel here was either stretched out by some amount or smushed in by an equal and opposite amount. Okay? So the nice thing is that displacement field is equal and opposite. The other source is what's called eddy currents. And the idea is that we talked about diffusion, we turn these gradients on and we turn them off. Okay? But as we're trying to change the magnetic field, because there's a lot of metal in the system, there's something that opposes that change, that causes the fields to change more slowly, and so the fields persist. Okay, so we change the magnetic field. Ideally, we'd like them only to occur when we're doing the diffusion gradients, but they can persist on into when we're doing the imaging, so they can actually screw up our imaging. Okay? That's shown here, where we'd like these fields in yellow to be done by the time we do our EPI. But when they last into that acquisition, they're going to change what our image looks like. Okay? And the problem is that, especially with diffusion tensor imaging, we're changing the directions of those fields every time, or quite a bit. And so that effect is going to differ from gradient direction to gradient direction. Okay? So we have to solve two things. We need to solve for susceptibility issues and also the eddy currents. Okay? And so what that means is if what we want to measure is this, what we actually get most of the time is something like that. Okay? And so we need to fix that before we pass it into the DTI fit. So one issue is with eddy currents, you can sort of see here, there's no diffusion gradients on, and so this is what the observed image looks like. Okay? Notice what happens when I turn on the X gradient. Okay? So there's diffusion weighting, but there's also slightly different distortion. If I turn on a different set of gradients, you sort of see not only getting diffusion weighting, but the whole shape of the brain is changing a little bit. And if I go to another set, it's changed a bit. Now, it's not huge, but it's enough that it's going to screw up your DTI fit if you don't take that into account. Okay. This is another picture from the book, which is showing that this is different diffusion directions, and you sort of see here, if you're looking at this ventricle, it's very close to this place here, and now it's displaced by quite a bit. Okay, so there's a lot of distortion that can go on. As we're changing the direction of the diffusion gradients, the shape of the brain also changes a lot. Okay. So what we have to estimate typically is we'll estimate one field homogeneity map first up, and we'll use top up to correct for that. And then for the eddy currents, we have to correct each volume separately. Okay, because that's changing with gradient direction. So how does top up work? It's actually it tries to estimate what the fields are. So first of all, it ends up it assumes that there's no field problems, and if it looks at the difference between the up, the top, and the down directions, and subtracts them, it ends up with a lot of problems, right? Because this image is very different from this image. So it just iterates and then comes up with another estimate, and these are more similar, okay, but not quite good enough. And it just keeps using that information to come up with a better estimate. And so here, it's fairly good, and then it stops when it thinks it's good enough. So it's basically this iterative algorithm that's trying to estimate the fields, and then for each field, it comes up with images and then corrects the images. Okay. And so this is just showing, before and then after, it's sort of corrected it quite well. How does eddy work? Well, it's really beyond the scope of this course to talk about how eddy works, but the idea is basically it sort of tries to it has a estimate of the eddy currents. Okay, it estimates what it thinks the problem is, and then it uses that estimate to predict what the images should look like. It compares them to what was actually measured, and then it updates the estimate. And so once again, it's iteratively trying to figure out what it think happened and then trying to correct for that. And it keeps going until what the prediction is and what's actually measured are the same. Once it has that prediction, then it can use that to correct the image. Okay. So, so what does that mean for in terms of your overall diffusion scan? Typically, you're going to have any number, let's say 100 diffusion weighted volumes, and then you'll have some other number of B equals 0 volumes. Okay, so typically like 6 or 8 B equals 0 volumes. So it will typically use the B equals 0 volumes to do the distortion correction with top up. Okay, because those have the most SNR, so that will get rid of that major distortion. Okay, and there's really two approaches that are done in the field now. The HCP takes the approach where it will acquire one scan with the phase encode one direction and another scan with the phase encode the other direction. So it will acquire two totally different distorted sets of scans. And then it'll merge those two and use all that information to come up with the best sort of estimate. Okay. The ABC takes a different approach because it's really starved for time, where it'll do some scouting scans at the beginning and then use that to correct the rest. So it won't acquire a whole data set in different directions. Okay. So once again, it uses the B equals 0 images and this is going to be used for the top up to correct that overall pulling and squishing. And then it'll use all the other scans to now also estimate what the eddy current distortions are. Okay. And eddy is still a work in progress. The FSL keeps improving it to take into account things like motion and other issues. But you should definitely be using something like top or eddy for any of your diffusion analysis. Okay. So I think that's actually the last slide. So we're actually ahead of time. So with that, are there any questions? Yes.
This may be a really, really dumb question, but are there tracks that connect with the ROIs, like can regions talk within themselves?
Yeah, that's actually a great question. So yeah, obviously it depends on how small your ROI is and how you define it. But and then there's also, certainly we know from neuroanatomy that there's lots of fibers and very tiny ones that we may not even be able to pick up with MRI. But there is more and work being done at going to higher resolution to try to pick up fibers, especially in areas that are mostly gray matter. Okay, so that is an emerging area, but it's not necessarily standard practice yet. Yeah, but it's certainly something. And there are, for example, there are lots of cases of like U fibers, right, which turn on themselves, and those can be very challenging for most methods. So there are people who are doing very high resolution and then also going to higher B values, really focusing on gray matter and other sort of more challenging areas. They are looking at things like that. But for these standard protocols and the standard analysis, it's not really quite there yet. Okay. Anything else? Okay. Yes.
Is it possible to have a tract make a kind of a sharp turn within a single voxel? Like I guess I'm wondering, would it be common for a single tract to turn within a voxel and then give like multiple diffusion directions?
You mean to make a bend?
Yeah, just make a bend like within a small voxel. Like, does that happen? Or
Yeah,
does
I
it
mean,
bend
no, I mean it can certainly happen. Yeah, and there again, it depends on the resolution, right? I mean, the fatter your voxel is, the more likely that is to happen. Yeah. So which is why people are always trying to, for DTI particularly, there is an advantage to going to higher resolution, right? So you can sort of, some of these issues, right, that you would have with a fat voxel, you can make go away with a smaller voxel. So it's not just the diffusion directions, it's also picking your resolution. And also, the more the less your imaging is susceptible to these sort of distortions and artifacts. So anything you can do to get rid of these also helps, because these all tend to smear things out as well, right? Because you're never going to be able to perfectly fix all those things. And so part of high resolution, high fidelity diffusion imaging is trying to solve these problems so that you're not ending up with this big blurry voxel that has lots of stuff in it. Okay. All right. Any other questions? Yes.
Are there advantages to removing physiological noise with diffusion imaging, or is that?
That's a good question. I actually don't know the answer. I'm not. If anyone knows the answer, let me know. But I haven't really seen a lot on that. I mean, certainly for fMRI, it's done quite a lot. For diffusion, I guess motion's a huge problem, and I guess a lot of the other problems are just so much bigger that. Now there is one issue where it is a problem, which is where it could have the brain is always pulsating, right? And CSF, and there's CSF flow, and so there can be at smaller B values there's more of that signal can last, right? And so, but for these higher B values, a lot of that stuff gets killed. So then it's not probably as much of an issue. Yeah, but if you're doing anything with like B equals 50, then there's still a lot of signal left that is affected by these sort of physiological fluctuations. So essentially, diffusion is sensitive to motion, right? But it's not just sensitive to diffusion motion, it's sensitive to any sort of motion. Okay, if there's no more questions, so that's it for today. So I will be out of town next week, so Dr. Simmons has Monday, and then Dr. Benema-Notstein has Wednesday. Okay.
And sorry, Frank's assignment will be due Monday, even.
Monday or maybe Wednesday, we'll see.
Okay, and we're not having like a 10 a.m. Friday session, or we are?
I have to check my email. I hope there is, but I'm not sure. But I'll let you know. Okay, thanks.